Welcome to the ORS Virtual Scientific Session, ORS Spine Section Celebrity Journal Club, organized by Dr. Simon Tang and Sarah Gulbrand. I am pleased to introduce today's session organizers. Dr. Tang is currently an associate professor in orthopedics at Washington University in St. Louis, and he's the associate director of the Structure and Strength Core of the Musculoskeletal Research Center at Washington University. The Tang Lab is interested in investigating the mechanisms of pain relating to intervertebral disc degeneration and inflammation in preclinical and clinical models. He is currently serving as the Spine Section Secretary. Dr. Gulbrand is currently a Career Development Awardee at the Philadelphia VA Medical Center and research faculty in orthopedic surgery at the University of Pennsylvania. Her research interests include the structure-function relationships of spinal tissues, imaging, and tissue engineering. She has been a member of the ORS Spine Section Membership Committee since 2015 and is currently serving as the Spine Section Membership Co-Chair. At this time, I am pleased to introduce Dr. Simon Tang.
Thanks for the introduction, Sherry. And I'd like to thank everyone for attending this first Celebrity Journal Club of the fall as a virtual scientific session. And I'm so thrilled to get together today so that we can discuss some really exciting science. Journal clubs are a common and effective way for groups of scientists to survey and critique contemporary and relevant literature. Typically, the trainees are the presenters to hone their presentation skills. The audience reads the manuscript ahead of time, so I hope all of you have read the manuscript. And the whole group discusses the study. However, the presenters are often asked questions regarding the experimental rationale that may not be immediately clear to the rest of the group. And so the resultant discussion often becomes speculative, even with the participation of experienced scientists. With the pandemic, a virtual journal club format allows the opportunity to promote and encourage scientific interactions and networking through celebrity journal clubs. So we can have the authors come in and provide sort of the behind-the-scenes insight to the experimental rationale in the study, and reduces the guesswork by the presenter. The authors also get a little more exposure and reaches a wider audience. So we're really excited about this format, and we've actually tried this a few times at WashU now, and it's been very successful. So we're really excited to share this with you guys today.
Thanks, Simon, for a great introduction to the concept. I'm super thrilled to introduce our two speakers today. So our literature review is going to be given by Sade Williams-Clayton. Sade is a fifth-year PhD candidate in the lab of Dr. Rosa Serra at the University of Alabama in Birmingham. She studies the involvement of TGF-beta signaling in the developmental regulation of spinal column formation. She's currently looking for a postdoc position geared towards musculoskeletal development and regeneration. And her fun fact icebreaker is that Sade is named after a famous singer, and she also sings as well. Our celebrity guest today is Dr. Rebecca Wachs. Becky Wachs is an assistant professor in the Biological Systems Engineering Department at the University of Nebraska-Lincoln. Her research focuses on developing immunomodulatory and neuromodulatory approaches for orthopedic pain. She's a native New Yorker and did her master's degree at RPI with Simon, actually, and she actually did her PhD in the same lab as me. So super thrilled to have Becky and Sade here today. And with that, I will turn it over to Sade to give her literature review.
Thank you so much for that introduction. I'm going to share my screen. Okay, hopefully you guys can see that.
Looks great.
Okay, good. Okay, so I really do want to leave some time at the end to facilitate discussion. So I'll try to get through this quickly, but not too quickly so that you understand what's going on. Okay, so today the paper that I'm going to be discussing is entitled Development of an In Vitro Intervertebral Disc Innervation Model to Screen Neuroinhibitory Biomaterials. You all know the author and you know who I am, so let's go ahead and go into the presentation. So what is the intervertebral disc and why is this model of innervation even needed? Well, so the reason that it's needed is because as we age or experience trauma, the intervertebral disc is prone to degeneration, and that degeneration can stimulate pain. And so what is the intervertebral disc? Just to give you a 3,000 view overview of this structure, I like to describe the intervertebral disc as the cushion of the spine. And this cushion has two salient parts. And so if you took a transverse section of an intervertebral disc and looked at it from a bird's eye view, you would see something that looked like this. So this pinkish inner region here is called the nucleus propulsus. It is gelatinous. It has a high water content due to the presence of a high concentration of GAGs or glycoaminoglycans, and it's very resistant to compressive forces. I like to call it the shock absorber of the IVD. And surrounding that gelatinous tissue is my favorite tissue of the spine, the annulus fibrosus. And this annulus fibrosus consists of a tough fiber cartilage. It has a very high content of type 1 collagen and very resistant to tensile forces. I like to call it the structural integrity of the IVD. So what happens as we age is that this tissue starts to break down. So IVD degeneration can manifest in many different ways, but the way that I'm going to show here in this diagram is when the annulus fibrosus, that outer encapsulation of the nucleus propulsus, it breaks down or thins over time, causes the MP to protrude out of its cavity, and this could cause protrusion on the spinal nerve, causing back pain. And again, that's just one way degeneration can happen, and that's just one way that chronic back pain can manifest. But there is another way that back pain can manifest, and that is due to the pathological innervation of these nociceptive nerve fibers, or these painful stimuli nerve fibers. So what you're seeing here on the left side of this diagram is a healthy disc. Again, let me orient you. So again, this green region is the nucleus propulsus, that jelly structure, and these outer rings are the annulus fibrosus, that tough fiber cartilage. What you can appreciate from this diagram is that these red little fibers are the vasculature and the yellow are the nerves. And what you can see quite obviously is that the IVD tends to be a very aneural and avascular structure, except for the very outer edges of the annulus fibrosus. So this is the normal endogenous structure of a healthy IVD. Now, if you fast forward in age, what you see is that upon a degenerative IVD is that microenvironment starts to change, and that it becomes infiltrated with vasculature and with these painful pain-sensing nerve fibers that cause the stimulation of pain. And so there's this change in microenvironment that happens, and this pain is debilitating because low back pain is a leading cause of disability worldwide, and it's a very, very expensive thing to have, with Americans alone spending over $100 billion each year on back pain. And currently there are no disease-modifying treatments. Currently the treatments are to treat the symptom, which is pain, and not to really modify the disease, which is stopping or slowing the progression of this degeneration. And to treat this pain, doctors usually prescribe NSAIDs, physical therapy, maybe an epidural shot if the pain is very severe, and the end-all be-all is spinal fusion. And so what this paper really wanted to get at is to develop a 3D in vitro innervation model to screen for neuroinhibitory properties of biomaterials to help treat discogenic pain. And so the thought process behind this is that if you can help modulate this pain by stopping the microenvironment of the IVD from changing from neuro-restrictive to neuro-permissive, and stopping that innervation of those pain-sensing nerve fibers, then you can help treat pain in these patients experiencing disc degeneration. So let's get into kind of the meat of the paper and some of the molecules that you will see mentioned in the paper. So their target molecule that they wanted to study for its neuroinhibitory effects is chondroitin sulfates. So why chondroitin sulfates? Well, as IVDs degenerate, their extracellular matrix degenerates as well. And the extracellular matrix of the IVD consists of a high concentration of these chondroitin sulfate proteoglycans, or CSPGs. And abertin is the most notable CSPG, specifically within the nucleus pulposus. And these CSPGs can inhibit nerve growth and vascularization depending upon species, neuronal cell type, and timing. And specifically for abertin, these chondroitin sulfates have two major sulfation patterns, which is chondroitin 4 sulfate and chondroitin 6 sulfate. And it has been shown that sulfation pattern can affect the neuroinhibitory properties of these molecules. And so knowing this paper wanted to address a couple of questions. Can chondroitin sulfate provide a robust neuroinhibition in this 3D model in vitro? And is this 3D model sufficient? Is it a sufficient way to screen for other neuroinhibitory factors in the disc? So with these questions in mind, I'll move in briefly into the methods, because the methods are incredibly important for this paper. And so the main method that you'll see throughout this paper is hydrogels, hydrogels, hydrogels. Just a preference, I am not a biomedical engineer. I'm not a physicist. I did my best in deciphering some of these things that are kind of alien to me, so bear that in mind. But the hydrogels, according to the methods of this paper, consist of methacrylic anhydride, biomaterials, and these other components that they throw in there. These two here in bold are highlighted because they're super important in testing these neuroinhibitory molecules throughout this paper. So the biomaterials in the hydrogels consist of these three things here. So you'll see these acronyms throughout the paper. So MAHA is methacrylic anhydride with hyaluronic acid. MACSA is methacrylic anhydride with chondroitin 4 sulfate. That would be known as CS-A from now on. And MACS-C is methacrylic anhydride with chondroitin 6 sulfate, which will be known as CS-C from now on. And so basically MAHA will be the control gel throughout this paper. It is controlled because hyaluronic acid is expressed throughout the disc, and it is known to not have any, it does not affect nerve fiber growth whatsoever. And so chondroitin 4 sulfate and chondroitin 6 sulfate will be the neuroinhibitory molecules that they'll be testing throughout the paper to see if they can modulate nerve fiber growth. And these are just a list of a few of the other methods and techniques they use throughout the paper. And so with that, I'll transition into figure 1. And so this figure was really just to set up, again, the methods of this paper. So panel A and B show the chemistry of chondroitin 4 and chondroitin 6 sulfate, which is again the neuroinhibitory molecules used throughout this paper. Panel C shows the isolation of a dorsal root ganglion from neonatal rats. This is how they isolated how they will test the nerve fiber outgrowth once they embed these dorsal root ganglions into the hydrogels. Panel D depicts how they make the hydrogels and how they embed either dorsal root ganglion cells or NP cells into these hydrogels. And so essentially what you see here is the construction of the hydrogels with either MAHA, hyaluronic acid alone, or MAHA in combination with either chondroitin 4 or chondroitin 6 sulfate. So this first upper panel here shows how they embed either dorsal root ganglion cells or NP cells into the hydrogel. They incubate and then express or shine UV light on the hydrogel to cause it to form, and then they culture for 5 days. And after culturing, they image these cells, they fix an image to view if there has been any neurite growth. Now this bottom panel here is the full 3D in vitro model of them testing the neuroinhibitory effects of either chondroitin 4 or chondroitin 6 sulfate. So what they do here is they pour the gel, let that sit, and then they embed the dorsal root ganglion into what they call the outer gel. And so this outer gel is permissive because it only contains MAHA, so hyaluronic acid alone. And then in this inner gel, what they'll do is pour in hydrogel that either contains the chondroitin 4 or the chondroitin 6 sulfate, and this is to test its neuroinhibitory effects. And so again, they incubate, cross-link, and then culture, and then view if there has been any neurite growth and image here. So panel E is showing basically how they're going to quantify if a hydrogel is neuropermissive or not. And so they're going to look at radial extension of the embedded dorsal root ganglions. They're going to trace the length of the neurite growth. And once they look at the hydrogel interface, which is this experiment here, saddling beside each other the permissive and non-permissive hydrogels, they're going to test here whether the dorsal root ganglion can extend into the inner gel via radial extension or trace length. Okay, so moving on to panel 2. Panel 2 is about testing the hydrogels. So panels A, B, and C is NMR. And this NMR is to check for the degree of methyl acrylation of these hydrogels. And so I am not an expert in this, but essentially this black arrow is the peak for methyl, for the methyl acrylate, and the white peaks are the peaks for either hyaluronic acid, CSA, which is chondroitin 4, or CSC, which is chondroitin 6. And the paper says that they basically measured the interval of the ratio between methyl acrylate and the biomaterials to show that these hydrogels have been well methyl acrylated. Okay, so panel D is testing the hydrogels for their stiffness. And so what you see here is that this is an oscillatory rheology machine. I did not know what that was, so I had to look that up. And rheology essentially is testing the stiffness of a hydrogel. And oscillatory just means the type of motion. And so basically, they use this machine to test the storage modulus, panel E, and the loss modulus, panel F, of these hydrogels. So I had to look this up. Storage modulus essentially tests the stiffness of the hydrogel, and then loss modulus essentially tests the manner in which a material dissipates energy. So these are just different ways to test different parameters of a hydrogel. And so what they tried to, or what they have established in panel E is that in this top line here, that's burgundy, this is the hydrogel with chondroitin 4. This second line is the hydrogel with chondroitin 6. This orangish line is the hydrogel with only hyaluronic acid. And this dotted line is porcine NP tissue. So what they're showing here is that the hydrogels containing chondroitin 4 and chondroitin 6 are significantly more stiff than those containing only hyaluronic acid and the native porcine NP tissue. So they show that even though that they are statistically significantly more stiff, that they are still on the same, basically, they're still on the same ratio, if you will, of scale. And so that is not a huge difference between native tissue and the formation of these hydrogels. So what they wanted to show here in panel F with the loss moduli is that they couldn't really get much information from this because there was just no discernible statistical difference between the hydrogels with the chondroitin, the hydrogel alone, or the NP tissue. So this essentially just established that the hydrogels are well methyl acrylated and that the stiffness of the hydrogels increased with the presence of the chondroitins versus hyaluronic acid alone. Okay. So now moving on to testing. Okay, we know that these hydrogels form and that they're well methyl acrylated and that they're stiff, but are they viable? Are the cells that are embedded in them, are they viable after extended periods of time? So what they did here is test the viability of NP cells. And they specifically test NP cells and not dorsal root ganglion here. It's because they anticipate this to be a therapy to mediate pain. So you would actually have to embed these hydrogels into the region of interest. And so they wanted to make sure that if you were going to do that, that the cells would be viable. So in panel A, what you see here is that they're testing for the metabolic activity of the NP cells embedded into this hydrogel by looking at Alamar blue. And so what we see here is that this dark green is hyaluronic acid alone, this light blue is chondroitin 4, and then this lighter green is chondroitin 6. And what they show here is that the metabolic activity each day in the individual conditions increases, meaning that these cells are viable and they are metabolically active each day. But if you compare the same day across conditions, there is no statistical difference. So essentially, this is showing that the cells are viable even after extended periods of incubation in these hydrogels. So this is another way to test cell viability. This is a live dead stain that they did. Again, hyaluronic acid alone, chondroitin 4, chondroitin 6. And what they say here is that chondroitin 4 has more live than dead cells when you compare it to either hyaluronic acid alone or chondroitin 6. And so again, the cells are living and they are viable. And these panels C, D, and E are just essentially a fluorescence verification of that. I guess the one question that I had, and I'll ask this at the end just to keep moving, is that it just simply, it seems here that there are just less cells in panel D than the other two, and I was just wondering if that affected the live dead percentage at all. But I guess just for time's sake, I'll ask that at the end as well. Okay, so moving on to figure 4. Okay, so figure 4 now is the money figure, I like to say. And so this is actually testing, can neurite growth be modulated in the presence of these chondroitins? This is the figure that we've been waiting on. And so what we see here in panel A is hyaluronic acid alone, and then panel B and C, chondroitin 4, chondroitin 6. What we see here is that there is extensive neurite outgrowth from this dorsal root ganglion in hyaluronic acid alone. We expected that. Hyaluronic acid, again, does not modulate neurite growth. But in the presence of these chondroitins, there seems to be a reduction of these neurites, especially in chondroitin 4 and some in chondroitin 6 as well. So D and F is just a quantification of that by measuring in D, neurite length, and in E, radial neurite extension. And you see here, it is statistically downregulated in chondroitin 4 compared to hyaluronic acid, and both are downregulated for neurite extension. Okay, so the next figure I'm going to show you is a figure from the supplemental, and I think that whether it was the reviewer's idea or their idea, I think it was an important thing to show. So this supplemental figure wanted to make sure that the change or the phenotype that we were seeing, where there is less neurite growth in chondroitin 4 and chondroitin 6, was not due to the increased stiffness of these hydrogels. So in the literature, others had suggested that stiffness can actually modulate neurite growth. The more stiff, the less neurite growth. And we saw back in figure 2 that the hydrogels containing chondroitin 4 and chondroitin 6 were significantly more stiff than those only containing hyaluronic acid. And so to address that concern, the authors simply made the control hydrogels more stiff by adding higher concentrations of hyaluronic acid. And so the original concentration of hyaluronic acid in the control gels is 2.25 milligrams per mil. And so what they did was they used increasing concentrations of 2.5, 6.5, and 8 milligrams per mil of hyaluronic acid in these hydrogels to increase the stiffness. And they verified that in panel F. So you see that this is 9 milligrams, 7.5 milligrams, and 2.5 milligrams, and that as you increase the concentration, the stiffness goes up as well. And what they wanted to make sure here, again, was that the stiffness was not causing the phenotype of reduced neurite growth, but the presence of chondroitins was. And so what you see here is that yes, that as you increase the concentration, you increase the stiffness, but that does not affect neurite growth. They still grow the same or even better as quantified here in D and E. So 6.5 and 8.5 had increased neurite length as well as increased neurite extension. Okay, so moving on to the next figure. So we verified that chondroitin sulfates 4 and 6 reduced neurite growth, and specifically chondroitin 4 was most effective at reducing neurite growth. And so they proceed with that chondroitin specifically for the rest of the figures. And so in figure 5, they're now testing this complete 3D in vitro model where they have an outer core that's permissive, so hyaluronic acid only, which is where they embed moving around this thing, which is where they embed the dorsal root ganglion. And then they have this inner core that can have only hyaluronic acid or hyaluronic acid with chondroitin 4. So with only hyaluronic acid, you see that as expected, there is neurite outgrowth. But if you change this inner core to include chondroitin 4, you see that neurite growth is now restricted, indicating that chondroitin 4 is acting as a neuroinhibitory molecule in this hydrogel. And so again, quantifications, C, D, and E are just quantifications of that with reduced neurite length, reduced neurite extension, and reduced neurite count. And so they wanted to verify that it was specifically chondroitins that is causing the effect of being restrictive to neurite growth and not just something else in the hydrogel. And so in order to accomplish this, they then added chondroitinase to the hydrogel containing the chondroitin 4 sulfate. So what they do here is panel A, they have this 3D model set up again where this outer core, which is permissive, is embedded with the dorsal root ganglion. And as a control, they just embed the inner core with the control, which is hyaluronic acid again, and seeing that, yes, there is neurite outgrowth. Now they do what we saw before in the last figure. They put the inner core with chondroitin 4 sulfate and saw, recapitulate the results. Yes, there is restrictive neurite growth, or almost none of the neurite growth now. But now what if we digest that chondroitin? And what we see is a small rescue of neurite growth into this inner core. And this kind of is proof of the pudding that chondroitin 4, specifically in this hydrogel, is restricting this neurite growth. And this is, again, quantifications of all of that. So neurite length is reduced in the presence of chondroitin 4, but it can be rescued. Same thing with radial extension, reduced but can be rescued if you digest that chondroitin 4 in the hydrogel, and same with neurite count. Okay, so now that leads me to conclusions from this paper. So conclusions state that chondroitin 4, or known in this paper as CSA, is a candidate to be used in treating discogenic pain caused by pathogenic innervation. The model is novel because it mimics the interface between this tissue, the inner core, and the external environment with sensory neurons, which would be the outer core in this model. Components of the 3D hydrogel model reflect the complexity of the ECM, or the extracellular matrix of the disc tissue, more so than traditional 2D models that may not have, the collagens, the laminins, the hyaluronic acid that these models have, as well as kind of the outer and the inner core. It includes all of those components. And they do mention that other variables involved in disc degeneration, such as pH, inflammatory cytokines, signaling molecules, would need to be added to make it more reflective of the disc microenvironment during degeneration. So a couple questions that I had, just two, was that I am a diehard annulus fibrosis studier, and this paper focuses on the NP, but axons must first be permissible through the AF before they reach the NP. And I was just wondering, does this model hold true for AF cells as well? And is this proposed model of therapy insertion of the hydrogels into the damaged disc, or the sole administration of chondroitin 4 alone? Is that sufficient? Does it need the hydrogel? And how does this hydrogel fare under physiological conditions if you were to insert this? And with that, I'll take any questions or comments.
So maybe to start things off, Shade, we can have you go back to that slide just to get things started, and we have quite a few number of questions that we're gonna I'm gonna go and cycle through. So maybe we can have Dr. Watts go ahead and start engaging in the discussion related to these questions.
Sure, I missed the early part, but great job on the last 3/4. It was a great review of it, so I'm excited. As far as the first question here, I completely agree. Right now, probably in some of you young healthy patients, the axons are really only in the outer annulus, right? Maybe, and as we age and degenerate, they can grow through the AF into the NP potentially. And so we have been playing around with this model as a potential for also testing materials for AF. So we have some collaborators that are interested in creating materials for AF that are neuroinhibitory. So can we test those sort of plugs in the central core of our model to actually screen for them? Are they also neuroinhibitory? Because I agree that they're going to have to get through the AF to get to the MP. And so the people that are developing some biomaterials for AF repair are also interested in maintaining that property. So when you say, does this model hold true for AF cells? I guess the one thing that we would want to do probably is for those people that we're collaborating with, we'd also want to test their materials to make sure that they are, cytocompatible with AF cells. But I'm guessing that they probably would be doing. Second question, is the proposed mode of therapy insertion of the hydrogel into the damages? So I saw Lori's question too about how are you going to get a UV cross-linked polymer into with a needle. So our proposed therapy, this paper was focused on development of a model. So we were looking at chondroitin sulfate as a screening tool for the model. And we do have other formulations of the chondroitin sulfate, that microparticle formulations that we could administer through a needle. We've just started to look at those and to see if they are also neuroinhibitory, right? So that we wouldn't have to worry about the UV cross-linking. An alternate thing we could also do is, right now we're methacrylating the chondroitin sulfate so that we can UV photoinitiate, but we could also add thiol groups to them, and so then they would be oxygen polymerized in the presence of oxygen, so then they would be injectable. So that's something that, and we have, yes, Lori, we have done that too. We played a little bit around with changing the crosslinker. Some of them aren't as cytocompatible, so we've with Lori just said that we could keep the methacrylation but change the crosslinker from UV to we've used AP and TEMED currently, but they're not terribly cytocompatible. So yeah, for sure. If anybody has any ideas of alternate crosslinkers for cross-linked methacrylate polymers that would enable injection and then gelation upon mixing after inside the disc, that would be great, because that would prevent a lot of rehashing of this. And then I think your question here, Sade, was about, is the chondroitin sulfate alone enough? And we're really still probing that question. We played around with a few different concentrations of the chondroitin sulfate, as well as the different, obviously, compositions, so CS4 versus CS6. And some are more potently neuroinhibitory than others. Our other work is in decellularized tissue scaffolds, and so those inherently have intact chondroitin sulfate proteoglycans, so they have some neuroinhibitory properties. So could we combine, some of these with that? Are they enough alone? That's an excellent question, and I think we would need to really test in an animal model before we could totally answer that.
There are a couple more questions. People are very active and curious, so I'll just go ahead. I guess you kind of got at the preclinical model, but I don't know, Becky, if you can specifically just discuss the rationale behind using UV cross-linking. If it is, how would you translate that in an animal model? Yeah, and so I think I know Lori had a question about that as well. We don't intend to, obviously. So the UV cross-linking is a function of the methacrylate groups that we have on there. So we use ErgoCure, which is a photoinitiator, which actually allows the hydrogel to cross-link in the presence of UV light. But there are other types of initiators that you can use, some of which are chemical in nature. And so we have the potential of using those if we wanted to have this be, essentially we could have a mixing needle that wherein the chondroitin sulfate would come in one needle, and then
the agent, the cross-linking agent, would actually come in the other, and then upon injection, it would form crosslinks. Some of those are a little bit cytotoxic, so we've played around with other formulations where we could add thiol groups, which under the presence of oxygen would cure or form a hydrogel. So that would be something where it would be just kept under nitrogen until it was injected and then be used. But that's sort of a different arm of the work, but I'm totally open to people have ideas about what we can do there or ways we can do that. And then obviously different types of formulations like particulating it or creating microparticles or nanoparticles that could just be injected.
So there's another follow-up question that's sort of related from Nicole Magotsky. The hydrogels used for the mechanical testing were cross-linked for 2.5 minutes per side, while the hydrogels used to assess neurite growth was only UV cross-linked for 1.5 minutes. How does this alter the mechanical properties of the hydrogels, and how do we reconcile the hydrogels used on the cells versus the one for the NP tissue?
So I think my postdoc, who's now works with Bailey, is on. I don't know if she's allowed to talk, but I know. So Sarah, the first author on this paper. So I will say I know that the hydrogels, the necessity for that became from the fact, like when we translated from the gel within a gel, the height of the gels are much smaller. So the UV light penetrates very quickly, cross-links the whole gel and creates a sturdy gel. But for the rheology, we actually use a punch gel. So it's a mold. So it's a much taller gel, and it's made between 2 glass slides. So the UV doesn't penetrate as well. We actually have glass between it. So we actually have to UV from both sides to get a robust gel. I think Sarah did some comparisons of like UV transmission. I don't know if she's
here.
Can you have her? Oh, there she is.
Hi. Yes.
Hey.
Yeah, you got it correct. I tried out several different UV times before we actually started the rheology, and what we really needed was thorough UV cross-linking in order to get the larger gel to stay intact, just based on the surface area and the volume and the ratio of the two. So that was just the basis for that.
And with regards to your question about sort of translation to actual properties of the gels, I think our goal here was to look at relative differences between hyaluronic acid and chondroitin sulfate in the gel. So we wanted to make sure that parameter for rheology was kept consistent, but these aren't necessarily the exact same properties that would be in the 1.5 minute cross-linked UV gel, if that makes sense, but pretty darn close.
Yeah.
We have a question from Nielsen Hogwin. Do CS4 and CS6 regulate specific types of neurites?
I don't think the answer to that's known. That's a great question. So there is a great paper by Ryan Gilbert from like 2005, I think. So he's a professor at RPI now. I think it was when he was in Ravi Bellamakonda's lab. It's a really early paper that looks at different types of CS and their regulation in neurite growth. I think it's an alginate hydrogels. And there are different types of CS that have different amounts of sulfate groups. So for example, there's CSA, CSB is actually called, I think, dermatan sulfate. Then there's CSC, CSD, and CSE. And I know for sure E has 2 sulfate groups, but I think A and C only have 1. So there is data to suggest in some systems that the dual sulfation actually does enhance, enhance, I guess, neuron inhibition. But it's not clear cut. So it definitely depends on the animal model or where the cells are harvested from. And the developmental age. So like for pup versus adult versus, neonatal or versus embryonic, there are differences in how those neurites react to the presence of CS. So I don't have a clear-cut answer. We're for sure interested in that. We also would like to, preferentially develop, in an ideal world, materials that are only going to inhibit nociceptors, right? We want maybe there is a need for some other types of nerve types, like maybe we want to let proprioceptors grow, right? So we have some idea of where we are in space, but we don't maybe want the painful nerves to grow in. And so ideally we'd be able to identify some biomaterials that could differentially modulate fiber type or, from growing or not. But right now, no, I think there's just kind of this yes or no of different CSs with an idea that like CSE should be more potently neuroinhibitory than CSA. Turns out it's really hard to purify and there's no commercial source for it anymore. So we've never actually tested CSE. The one guy that used to do it just retired and he said he would send us his stock. He's in Japan. But as of yet, we haven't tested CSE.
Cool. So the next question is from Nadine, which is, what is the role of the negative fixed charges in permitting or inhibiting neurite outgrowth?
Great question, Nadine. I don't know that I have a good answer for that one yet. We've just started to play around with the role of charge in our microparticles and trying to understand the role of charge in not only neuroinhibition, but also, binding of growth factors or drug release of other things. So ideally, if it does play a role, I think that could be something really interesting to look at, right? If we could control the charge and or increase or decrease, but we have not yet looked at that. I'm going to start taking notes here. This is great.
So our next question is from Nicole again. What do you think would happen, Becky, if you had a hydrogel that contains both CS4 and CS6?
Great question. I mean, I should throw that one to Sarah. She's looked at a lot more of these gels than I have. I'm guessing that it would be, I don't know. I don't know if there's kind of a maximal, that there's a point of diminishing returns, right, where the CS alters the matrix in other ways and then therefore doesn't maybe have the same neuroinhibition. Because I know when we do add more CS, we do stiffen the matrices. And so that could be an undesired change. So, but I would think there probably would be some degree of increased neuroinhibition to a point, but then there probably is some decreasing returns on that.
Yeah, I would say the same thing. I never tried mixing the two of them, but if we doubled up the concentration by adding in the same amount of both CSA and CSC, I think the stiffness would become a factor before the added chondroitin sulfate would.
Plus CSC was, I don't want to swear, this is being recorded, was really annoying to make.
That's the type of stuff we want to hear from these journal clubs. It's really this true rationale behind the experimental decisions, right? So
yeah, we made it. I think we did 3 whole batches because we wanted to make sure that we were seeing what we were seeing. But then it was like CSA is so much easier to make and it's more neuroinhibitory. So let's just go with that one.
So we have Lori next.
I do want to sort of throw it out there that I think the photo cross-linking was a really good idea for this system, because I know in the past when we have methacrylated things, you can get a lot of different groups that take up the methacrylate. And then if you throw in like a thiol chemistry or something like that, you'll wind up like cross-linking, everything to the native tissue, to the cells, to anything that's, available and reactive with the thiol. So it's probably a cleaner system to work with the UV. But I did want to know, why did you have to do initial thermal initiation? I've not seen that before. What was that for?
That was because we had collagen in the gel. So we actually wanted the collagen to form hydrogel, a base. So the collagen, so hyaluronic acid on its own is not permissive to nerve growth just because there's no binding sites. So there was collagen and laminin mixed in at low concentrations. And so the collagen actually forms a thermal gel before we UV initiate. So it just allows that to happen, because we found that once we UV initiate, the collagen doesn't form the fibers as well, right? Because you already have kind of a fixed space.
Okay, and then I guess sort of a second question was the choice to do chondroitin 4 versus chondroitin 6, like as opposed to throwing in like the whole range of, like heparin sulfate or keratin sulfate, or like have broader dispersity, right, in the species that you were investigating here, because it's hard to understand why the sulfation pattern specifically is going to regulate the system versus it must contribute to some conformation or chirality or something.
It actually directly binds, so it binds, the neurite binds like the chondroitin sulfate. So there's like PTP sigma and another one that I'm escaping. I did not prepare for this journal club, so this is all in my head right now. Actually directly binds chondroitin sulfate. So we picked, so I will say for the purists out there, I won't call it CS4 and CS6 because it is truly CSA and CSC, because the different types actually are blends and they're not pure. But anyway, so I think our CSA is from bovine trachea, is that, or is it Sarah, you're out there?
It's bovine. It is bovine. I don't remember if it's trachea.
Okay. But anyway, so the reason to go with CS is that it's known in the spinal cord to be like the potent neuroinhibitor. And so there's been a lot of work into the different types. And so it's a good sort of starting place. With regards to heparin sulfate and keratin sulfate, they actually they're not neuroinhibitory. and so that's why we stuck with the CS side of things. And then honestly, with regards to types, Sarah can speak to this too, is that it's just really hard to get any more than those two. You can get dermatan sulfate, but it doesn't have as much neuroinhibition. I think that's CSB. But so really that's why we went with the ones we went with.
Simon, may I say something?
Yes, Mara, I was just going to call on you.
Okay. Thank you. So I think here we are doing an academic exercise because the reality is totally different. You know, in the 80s, Japanese people mainly, they were using protoglycan and chondroitin sulfate to induce pain in the spinal canal by adding the protoglycan, mixture of protoglycan, I'd say. And so I think we are really looking at an artificial system here. And I would like to stress the fact that mainly there is no protoglycan, or much less in the annulus fibrosus, which it's maybe a strong suggestion that you maybe have ideal system where you just take the nucleus pulposus situation, even more artificial than the nucleus pulposus situation, to build your system. So I think you maybe want to have. I can suggest that maybe take some explant of annulus tissue and use it to in your system to see effectively if you have first degenerated and not degenerated, to see if your nerves are growing or not, and then you can use your system to study some inhibitory factor or whatever. That's my, that's 40 years ago. There is a lot of literature on Japanese people mainly, but also from, I guess, Chicago or and the old time San Francisco, where they did this type of work of putting protoglycan extract, mimicking a little bit the herniation of the disc, where you get extrusion of the nucleus and you get inflammation. So maybe you can refine your system by using explant of annulus, taking maybe from different patient and so on. And so that was my main concern of all, to sell it as a 3D model for innervation and study that. It's there, but I think we still have a lot to do, and maybe to modify the system during the process. That's one thing. And then I had some other question about the system, but I forgot about that. That's fine. Too long.
I'm sorry, it's a little while, but thank you, Mara, and just for everyone who doesn't know Mara Alini, he's also one of the editor-in-chiefs for. Okay, we have a few more questions, so I think we have just enough time to make it through all of them. So the next question is from Nafrika, who asks whether or not the hydrogels are isotropic, and if not, how do you, or how. If they are, how do you assure it?
Yeah, I mean, I think I can say yes, but it's not an assured thing. We haven't tested it. We haven't looked at any sort of distribution. I mean, I don't think we've done some where we've mixed in particles and looked at their uniform distribution within it, just to show that like our mixing is complete, right? That we've actually uniformly mixed. And I guess Sarah could probably speak to this a little bit, but I know that when we make the gels, there's 10x DMEM in there, and so this is totally not an assured isotropy. But you make sure the pink is uniformly pink, right? If you're mixing hydrogel in with a solution, and so I know it's really difficult to mix, and so we have some what are they called? The pipettes that things don't stick to?
Low retention.
Low retention. We have low retention tips that help with the mixing of these thick gels. But no, we haven't in any way verified that they're isotropic. But based on the robust growth we see of the neurites in all directions, we assume that, at least for the things that encourage neurite growth, that it is isotropic.
Next question is from Jordan. Jordan's question is, why does NP break into the AF over time? Is it being loaded in some way or pre-strained by the AF?
I'm not sure I'm the best person to answer this. I mean, there's biomechanics people out there, but from my understanding, there's a couple different reasons that the NP, so one of them is like an acute herniation, right, where that could actually be sort of a tear in the AF that allows the NP to, push through and herniate out. And that's likely due to some overloading or, a car accident or something that's going to cause some acute overloading. But then I think when it comes to like age-related degeneration, there's just kind of a loss of so as the NP becomes more fibrous in nature, it shifts kind of from type 2 collagen to type 1, loses some of those proteoglycans. The actual sort of region between the NP and the AF just starts to get blurred. It becomes less of a gelatinous kind of highly water-filled and sinks more into a compressive state. So I wouldn't say that it necessarily pushes out into the AF, but more that the distinguishing line between the two is no longer as clear cut. I don't know if anybody else, feel free, people that actually study that sort of thing to chime in.
Yeah, I think one of the hallmarks of degeneration is the loss of that border, the annulus nucleus border, and so as that kind of differentiation starts to deteriorate, then you also start to have the NP start to creep into the AF. So the next question, or next comment rather, is from Ryan Gray, which is actually pretty clever. So Ryan Gray suggests that you could most likely cross-link the hydrogels using optical fiber, essentially at the same time as the needle delivery. So you just do it all in one go and still be able to control the effects of cross-linking. So that's a very cool idea. Sade and Becky both did an amazing job, and the discussion has been really lively. I was just going to say, I guess one thing I can say is I feel like from our lab's perspective, one thing we've really pushed for, this is my mantra, so for whether it's true or not, I don't know, but our lab is focused on trying to get towards a more painless degenerative state. So instead of pushing to try and regenerate the disc, can we actually discern what are the different components that actually modulate nerve growth and pain independent of degeneration? Because it is such a multifactorial process, if we're not going to, restore nutrients to the disc and we're not going to be able to sort of shift a lot of the cascade that's already going on, can we sort of decouple them? And so that's kind of where the motivation for this paper came from, was that what if we just look at the agents that are lost with time and age in the disc, like aggrecan and these chondroitin sulfates, and can we actually mimic them independent of, rejuvenating a type 2 collagen with, all the healthy proteoglycans and, sort of decouple them. And so, so that was just, I missed the beginning, maybe that was totally said, but that's kind of the direction we're pushing into as a lab. So if you're interested and want to come join our lab, feel free. That was my plug. Well, this is a good avenue for it. I thank everyone again for participating and engaging in a lively discussion. We hope to do it again. We'll figure out the best avenue to do it, but especially tomorrow. I don't know what time it is for you, but thank you for calling in.
Very good. I congratulate the SAID. Very well done analysis of the paper, and Becky also for starting this important line of research on pain and intervertebral disc. And hopefully we will, you will help us, the GR Spine, to succeed and become the home of all your effort and work. Thank you.
All right, thank you everyone. We're right on time, so thank you again.
Thanks everyone.